And what basically is a cloning vector? It is basically a circular plasmid. We talk about what it is a circular plasmid, right? As you can see. And these colors, these are just a DNA. It's a DNA. It's a circular DNA. Now, everything that you are seeing here are names of restriction sites. Okay. So I told you about restriction sequences, right? Restriction enzymes. Can you tell me quickly what are restriction enzymes?
They are molecular scissors.
Yes, restriction endonucleases I'm talking about. They are called REs, or as very well pointed, they are called molecular scissors. Why they are called molecular scissors? Because they can cut a double-stranded DNA at specific locations or at specific sequences. Is that part clear?
Yes, sir.
So HIND3, BAM-H1, SAL1, PVU2, ECO-R1, CLAV1, PVU, PST. All these are restriction sites. So these are the sequences where the restriction enzyme with the same Yep. Hello. Am I audible, people?
Yes, sir.
Yeah. I'm so sorry. I got a power cut at my place. So take some time. Okay. Back. Can you see everyone the screen?
Yes, sir.
Yeah. So I was telling you that these are plasmids, plasmid vector. Okay. Now we know that what plasmid vector are used for. It is a vector, a tool in which we can put our gene of interest, and we can send it in another system where it can get the gene can get expressed, and the gene product, which is the protein, can be synthesized. Now PBR3 this P stands for plasmid, BR stands for the lab in which it was first synthesized, like the lab which formed it. So it's a synthetic vector, as we know. It's artificially created with all these sites present. So this is it's like kind of a ready-made plasmid for you. You can just order this plasmid, get it for your research work, and you can decide which restriction endonuclease. So these are BAM-H1. So the BAM-H1, and I told you how these are named, right? EcoR1, E. coli, R strain, the first endonuclease from this category, right? HIN3, Haemophilus influenza, DS strain, the third restriction endonuclease, which is discovered in this system, correct? Similarly, PBR, P is the plasmid, BR is, I think, it's Bolivar and Rodriguez lab from where it came. 322 is the number, so don't worry about that. Now, this plasmid has few things which are important. First is this ORI, this region. So these are all sequences of DNA. So this is just a representative of the plasmid. In reality, a plasmid is a DNA sequence, a DNA thread, which looks something like this. It is a double helical coiled DNA, which it just have to be circular. You understand? There
Yes.
should not be any open end. And from this point to this point will be one sequence, A, T, G, C, something like that. So this is ORI, which is origin of replication, right? From here, the plasmid starts replicating inside the cells. So the direction will always be like this. If the ORI 3' to 5, 5' to 3', so direction will be this. Then immediately after ORI, in this is your ROP. Now ROP is this region, which they have, I think, mistakenly marked to this green region. This green is PVU. So this is ROP. It is called the region which contains proteins which are important for the replication. Okay. So replication starts here, and then first the replication, transcription, and translation of the proteins will happen that will help this process again. Then everything that you see after that in a sequence is a restriction site. Okay. So by using these endonucleases, you can cut this plasmid as per your wish anywhere. So if you want to use SAL1, you can cut the plasmid here. BAM-H1, you can cut the plasmid here. HIN3, you can cut the plasmid here. EcoR, you can cut the plasmid here. Now, why do we cut the plasmid? Tell me. And we only create one cut. Why is that? Maimuna, Samara, why do you cut the plasmid? We cut the plasmid to just insert our gene of interest. Remember, we will make one cut, it will open up, we will insert our gene of interest. So that's why you will see that every restriction endonuclease has just one site. PST has just one site, PST1. PVU1 has one site, PVU2 has another site, because they are two different sequences. In a plasmid, you will never find, for the same restriction nucleus, there are being two sites. So if there is BAM-H1, there cannot be another BAM-H1 site here, because that will just make the plasmid useless. That plasmid we can't use. Make sense, everyone?
Yes.
Samara? Yeah. So now do you understand what is this?
Yes, sir.
Third thing is, you see this AMPR, TETR. What are these? Let me read. Yes. So what are these TETR, AMPR, and yeah. This is, remember we used marker, the marker region. Yes. A marker sequence that will tell us whether our plasmid is transformed or not, whether the organism in which we are putting the gene has our gene of interest or not. So in this ready-made E. coli cloning vector, there is already a cassette of, like, a gene sequence from this region to this region. This whole region, which is shown in pink, and here this whole region, which is shown as in pink, is the sequence which provides you resistance against ampicillin. Ampicillin is an antibiotic, so this is ampicillin resistance. And here this TET is tetracycline resistance. Again, it is an antibiotic. They are different antibiotics, but they give you two cassettes, two sequences, one giving you ampicillin resistance and other giving you tetracycline resistance. Now let's say you used BAM-H1 as your restriction endonuclease, because this suits your gene of interest the best. And remember, I also told you the restriction endonuclease that you will use to cut the plasmid, you have to use the same restriction endonuclease to cut out your gene of interest. Remember that?
Yes, sir.
And why is that, Samara?
To create the same sticky ends.
Perfect. So if I use BAM-H1, okay, if I have not entered, if I have not inserted any gene of interest, let's say I just ordered this plasmid and put this plasmid as it is in a E. coli bacteria, or any, like, a bacteria that I'm doing an experiment on. If I just give this raw plasmid to that bacteria, so that bacteria should grow on a ampicillin plate and in a tetracycline plate both, right? Because it has resistance against both. These sequences are intact, correct?
Yes, sir.
That is called testing the plasmid. So whenever a lab orders in research, we do that. So we first test the plasmid. To test the plasmid, the vector contains a resistance site already. Now we just have to make competent cells. Remember, I told you about making cells competent by using divalent cation, gene gun, etc., etc. So we make cells competent, bacterial cells competent, so that they can take up foreign DNA. So this plasmid is a foreign DNA. Then we transform the cells with this plasmid. So these plasmids enter inside. We give them heat shock, right?
Yes, sir.
And these plasmids enter. Now, just to check that plasmid is functioning properly as a test, as a control, we will take those bacteria which we have transformed and put them on an ampicillin positive plate, culture plate, and a tetracycline positive culture plate, or a mixture of both. And they should grow. if the plasmid is working perfectly, right? Because if the plasmid is working perfectly, ampicillin resistance gene is getting replicated, transcripted, and translated to produce a protein which will help the bacteria to grow on an ampicillin plate, basically giving the bacteria ampicillin resistance. Make sense till here?
Yes, sir.
Now there are two sites, and you will see that in this site there is BAM-H1 and SAL1. In this site there is PST1, PVU1 sites. Now if I'm using BAM-H1 as my restriction endonuclease, it should cut my plasmid here, right?
Yes, sir.
And my plasmid will open from here. Now half of the sequence will be there, half of the sequence will be there. In between, I will insert my gene of interest. Okay. So now, if I transfer this plasmid into an E. coli and test whether my gene of interest is there or not, then if I grow my transformants in an ampicillin plate, they will survive, right? Because they still have ampicillin gene there. But if I grow them on a tetracycline plate, will they survive?
Sir, can you please repeat?
Yes. I cut the tetracycline resistance gene in between using BAM-H1, right? To insert my, let me clean all of this. No, not this. This should stay. Everything is clean, okay? Just focus on these green regions. So from this point to this point, this is a sequence for tetracycline resistance gene, this one. Make sense? TET-R. And this is a sequence for ampicillin resistance gene, correct?
Yes, sir.
When I have not used any restriction endonuclease to insert any gene of interest, and I have taken this plasmid as it is and inserted it inside a E. coli, just to check if my plasmid is working properly. By working means the plasmid is replicating, transcripting, and getting translated. So to test this, if I grow these bacteria now on ampicillin plate and tetracycline plate, they should show resistance and grow, correct?
Yes, sir.
Yeah. Now, the question is, I used BAM-H1 as my restriction endonuclease to cut my gene of interest. So I will use the same to cut my plasmid as well, correct?
Yes, sir.
But when I used BAM-H1 to cut my plasmid, what did I ended up cutting? I ended up cutting the gene which was giving the bacteria tetracycline resistance into two parts. And in between, I will insert my gene of interest, correct?
Yes, sir.
So this gene, which was giving tetracycline resistance, now is useless. It will not function, correct?
Yes, sir.
Now to check, what I can do is, I can put my bacteria on an ampicillin plate and it should grow or not grow.
It will grow.
It should grow. But that does not mean that my gene of interest is properly carried. Now, because I have already done the control experiment, remember, where it was growing on both antibiotics.
Yes, sir.
If after inserting my gene of interest by using BAM-H1, if it still grows on ampicillin, but it stops growing on tetracycline, what does that mean? After using BAM-H1 as restriction endonuclease of my choice and cutting the plasmid at this place here, then inserting my gene of interest then putting back the plasmid in bacteria, and then again doing the same experiment where I tried to grow the bacteria on an ampicillin plate and a tetracycline positive plate. It grows in ampicillin, but it does not grow in tetracycline. What does it mean? It means that I have successfully been able to give my gene of interest using BAMH1, because then only the tetracycline now is inactive, because I created a cut and inserted something here. So this whole gene become non-functional. But say, this is still functional because I did not do anything to this. Make sense?
Yes, sir.
So this is how we use plasmids as vector. There are many such plasmids. This is one very, very standardized and very readily used plasmid. Even we were using. There is also a PUC19 plasmid. This is also used very, very much. It also has ampicillin resistance gene. It's called PUC19. I think P is smaller, UC19. It's called PUC19 or PUC19. PBR, PUC. There are many such plasmids, which are ready-made plasmids. Anything else, Sumaira, that you have a doubt? Sumaira, Mamuna, any other doubt? Or do you want me to repeat this part again? Or any part from this again?
Sir, like on page number 200, the first para.
Page
Can you
number. Yes, please. So let me open that. 200.
Yes,
Insertional inactivation you're talking about, right?
yes,
Okay, great. Yes, so I gave this portion as a homework to you to read, and we discussed it, but we can do it all over again, not at all an issue. Okay, so let's start with reading, or should I do one thing? Should I take this paragraph and paste it there? Yes, that will be better. Yes. So as I told you, there are two ways to use selectable markers, correct? To know that whether our plasmid has worked or not. Yes or no?
yes, sir.
Yeah. You all can see the screen still, right?
Yes, sir.
Okay. So let me paste the paragraph here. This you're talking about, if I'm not.
Yes, sir.
Okay. So would you like to read that, Samara, please? Should I increase the font? Should I zoom it more?
Yes, sir.
Okay. Sorry. Is this better?
Yes, sir.
Yeah.
Selection of recombinants due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneous plating on two plates having different antibiotics.
Yes, just stop here. So this line just tells you about the experiment that we just did today, just did right now, right?
Yes, sir.
So what did I say? When I order the plasmid, which has two antibiotic resistance genes already, I first have to do a test to check if the plasmid is working. I will make two plates, ampicillin, tetracycline. Put these bacteria containing the plasmid on these two plates. If they survive on both, it means my plasmid is working. Now, having done that, next I will use a restriction endonuclease. And if I have used BAM-H1, then I will again do the experiment. It should survive on ampicillin, but not survive on tetracycline because I have disrupted this gene, correct? So this is the whole way to know that my marker, selectable marker. So here I am making sure that my gene of interest has been passed on by inactivating the antibiotic resistance gene. So either I am inactivating this one by using BAM-H1, or I will inactivate this one by using either PVU or PST, correct?
Yes, sir.
But this seems to be a long procedure, right? First plate to know whether it's working, then do the insertion, then again plate to see if the inactivation of antibiotics has worked or not. Okay. And I have to, every time I have to make 2 plates having 2 different antibiotics. Sometimes I can also make 1 plate having both the antibiotics, but then I have to optimize their dosage and all. Lots of work, correct?
Yes, sir.
So is there any other alternative? Yes, there is. And instead of talking of inactivation of antibiotics, what we can do is this. So can you read again from here?
Yes, sir. Therefore, alternative selectable markers have been developed which differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of a chromogenic substrate.
Substrate. Yes.
In this, a recombinant DNA is inserted within the coding sequence of an enzyme beta-galactosidase. This results in inactivation of the gene for synthesis of this enzyme, which is referred to as insertional inactivation.
Correct. So what they are saying is, so here again, when we are making an antibiotic gene inactive, whether it is tetracycline resistance gene or it is ampicillin resistant gene, when we are making them inactive, we will only get to know after plating, after making a plate and culturing the bacteria, right?
Yes, sir.
So scientists thought, how about instead of this two-way system, can we develop a single step system where the moment I use something else instead of this antibiotic resistance gene, I use something else here in this pink region. And the moment I insert my gene of interest by cutting that part, that gene gets inactivated the same way it is getting inactivated here. But this time, I don't have to plate. I can just get to know on the same plate where I'm growing the bacteria that if my inactivation, if my gene of interest has been properly placed or not. And how? By a color change. So suppose you are growing your bacteria and the color of the plate is yellowish or greenish, let's say. And then these are the bacterial colonies growing on it. And when this insertional, when this inactivation of the gene happens, you will see that in the vicinity, there will be a color change. Like there will be green will start turning yellow. And you can now figure out that, see, for these colonies, there was a, so you can say for this colony, 1, 2, 3, 4, 5, there was a color change. But these colonies showed no color change. So maybe they are not carrying they are not transformed, but they are transformed. This is a better way, right?
Yes, sir.
So this is exactly what they have done. So there, chromogenic substrate means a substrate that has the ability to show color change upon reaction. So the alternative is to use a selectable marker which can differentiate recombinants, which means those bacteria which carry our recombinant DNA, from non-recombinants on the basis of their ability to produce color in the presence of a chromogenic substrate. Now in this, a recombinant DNA is inserted within the coding sequence of an enzyme beta galactosidase. So instead of ampicillin or tetracycline, we are using this beta galactosidase. Okay, is it clear?
Yes, sir.
I'm using this as a selectable marker. Now, how does it work? It works that when you inactivate this gene, okay, by inserting your gene in between. So this is called an insertional inactivation of beta galactosidase. So I inserted my gene of interest because of which the beta galactosidase gene got inactivated. And then I add a chromogenic substrates, okay, which will give me the presence of a chromogenic substrate gives blue colored colonies. Okay, if the plasmid in the bacteria does not have an insert. So when beta galactosidase is active, so beta galactosidase is active, which means I have not inserted anything. Then when you grow the bacteria, there will be blue colonies. Is it okay?
Yes, sir.
Now, if I have inserted my gene, the beta galactosidase is inactive. So when beta galactosidase is inactive, now when you, and how can it be inactive? There is only one way for it to be inactive. If I have inserted my gene of interest in between this, right? I have used a restriction endonuclease which cut beta galactosidase in the middle and insert my gene there. Now beta galactosidase is inactive. So they show no color. Cool. Does that make sense, Samira?
Yes, sir.
So when you order these plasmids which work on beta galactosidase insertional inactivation and not on antibiotic resistance, then to first check, you will put your plasmid vector in a bacteria without inserting anything. And you will see that all the bacterial colonies will be blue, those which are having your plasmid. You are happy. Okay, great. It works. Now let me insert my gene of interest and I insert it in between the beta galactosidase gene. So this get inactivated. Now all these bacteria which have my gene there, my recombinant DNA there, will show no color, will be colorless colonies. Okay, and colorless colonies growing on a agar plate looks like slightly whitish, but they will not look blue. So if on a plate you find any blue colony, you will not use it because these will be non-recombinants. Correct?
Yes, sir.
And if you find these whitish colonies, which does not have this blue color, these are the colonies which have my gene of interest. So I will use this one. I will not use this one. Correct?
Yes, sir.
So do you understand this paragraph? This is another way of using another selectable marker. And here I don't have to make two plates every time and check whether it is surviving in one, it is dying in the other, or is it surviving in both, or is it dying in both. Just one plate to test and the other plate to grow. And all the colorless colonies are mine of my use, which are containing my gene of interest. All the blue colonies are of no use to me because they have intact beta galactosidase, active beta galactosidase, which means they do not contain my gene of interest. Just, I hope it's clear.
Yes, sir.
Yeah. Just give me 1 minute, people. I need to drink some water, okay? I'll be back in a minute. Is it okay?
Okay, sir.
Yeah. Any other doubt? Please look and ask. from biotechnology. We are doing this chapter, so let's take any other doubt. Which Mamuna, do you have any doubt? I'll be back in a
No,
minute.
sir.
Yeah, I'll be back in a minute. Hello, am I audible?
Yes, sir.
Yeah. Yes. Yes, please tell me more doubts. Any other?
No, sir.
No, from no doubt from this chapter.
No, sir.
Any other chapter? So I'll be giving you, all of you, units that we will be covering. Okay. So I want to go through the whole syllabus once this month for important concepts. So this is biotechnology. Okay. So let's start with genetics first. So in the next class, I want you to focus on the genetics unit, chapter 5, 6, and 7, including evolution. Okay. And come up with any doubts that you have in these three chapters. Okay. Chapter 5, 6, 7. I think 8 is not in. Only 3 chapters are there in genetics unit, right? If I'm correct. Yes. Then comes human disease, health and disease, microbes. Oh, okay. So we will take chapter 5, 6, and 7 first for any doubts. And if you don't come up with your doubts, then I will start with important concepts. Okay. So in the next class, let me discuss with you a little bit what I will be focusing mostly in next class. So let's go to chapter 5, genetics. Okay. Let's start with our own notes. And what I want you to revise in the next class is this laws of Mendel. So all 3 laws of Mendel are Clear to both of you?
Yes, sir.
Law of dominance, segregation, and independent assortment. So here are some questions. First is, which of these three laws is also known as a pure law with no exceptions? Which of these three laws have no exceptions? Memna. Law of dominance, segregation, or independent assortment, which has no exception in nature?
Segregation.
Segregation. And what it is also known as? You're correct. It is law of segregation, which has no exception, and it is also known as law of purity of gametes. Okay.
Yes.
And Samara, what are the exceptions of law of dominance? What are the exceptions of law of dominance? Are you there, Samara? Excuse me, people. Yeah. I think she's not able to hear me, so it's you, Memna. So, Memna, would you like to tell me what are the exceptions of law of dominance, where this law is not followed?
Um,
Yeah.
I don't really know. Probably like the generations in the parents or something.
No. One is, incomplete dominance. So exceptions for law of dominance is where so you know what is law of dominance, right? In a heterozygous condition, so every gene has at least two alleles, right?
Yes.
Alleles are just different forms of the same gene, slightly different forms based on their sequences, where one allele can be dominant over the other, but the gene is for the same. It's the same gene. So the gene, let's say, that gives uh, uh, which is responsible for the height of the pea plant, capital T and small t. These are the two alleles, right?
Yes.
So capital T is always dominant, right? And this is the recessive one. So in a heterozygous condition, if there is a capital T coming from the mother and small t coming from the father, the two alleles, which allele will be expressed? Capital T. So this F1 will all be tall because small t won't be able to show its effect. It will only be able to show its effect in a homozygous condition. So here only there can be a small plant, right?
Yes.
So first, yes, Samara, you have joined. Can you hear me now? We are discussing the exceptions of law of dominance. So first is incomplete dominance, right? Which I think we discussed below somewhere here. Yes, exceptions of law of dominance. First is incomplete dominance, which shows blending of traits. So technically, if R is for red and is dominant, and small r is for white and it is recessive, okay, let's forget this for now. This is not important. So in a capital R condition, you will find and you expect that the flower should be red, and that's okay. In a small r, you will expect that the flower should be white because recessive, right? And that is also okay. But what do you expect in a capital R, small r condition? Normally,
Red.
a red flower. But instead, if you are getting a pink flower, now pink was not at all coded genetically, right? There was no allele giving a pink flower. capital R was giving red, small r was giving white. So either this red pigment is present or not at all present. And white is there. But instead, there is a blending now happening. So capital R and small r gives pink. And when do we get pink in a combination of red and white? When we mix both, right? If any one of you has painted, if you mix a red color with a white color, it becomes pink color. Make sense?
Yes, sir.
So this is telling you that law of dominance is not completely being followed here, and there is some cheating happening. And that cheating is happening due to blending of traits. So the only reason could be that this capital R is not able to show complete dominancy over small r. And small r, which should remain silent and non-expressive in the presence of capital R, is also showing its effect. And that we see in a snapdragon flower, the flower of the snapdragon plant. And this is where they realized that law of dominance, which was given by Mendel, does not hold true everywhere in the nature. It is mostly true, but it has some exceptions. And incomplete dominance is one exception. Make sense?
Yes, sir.
Mamuna?
Yes.
Yeah. So you have to revise these things, okay? And if you are not sure that you know it, please ask me. Ask me any number of times, not not a problem, okay? The second exception is codominance. So codominance is, again, it does not follow the rule of dominance, where one should always be dominant over the other allele. So this we see in the ABO blood group. And in the AB group, so there are 3 blood groups possible. Sorry, 4 blood groups which are possible. A group, B group, AB group, and O group. So for A, B, and O, there is normal law of dominancy followed. Okay. Because A can either be in the presence of allele IAIA or IAIO. Remember, I told you that O means nothing, right? O is nil. So there are only 2 antigens which can be present on the surface of RBC. Either it can be antigen A or antigen B for this blood grouping. If both are not present, we call it O. So if from one parent you get A, from the other parent also you get A, your blood group definitely has to be A, not a problem. It is like a capital R condition. Perfect. If you have A from one parent and O from the other, which means from other parent you got nothing. So clearly A will be dominant, right? Normal, again, no problem. Same with B. In case of O, there is no antigen, so your blood group will be O. But in case of AB, where you get antigen A from one parent and antigen B from another parent, so on your RBCs, when you start producing your blood cells, you have gene for both the antigens. So both antigens will get transcribed and translated, and these will be present on the surface of your RBC. So what do you expect your blood group to be? You will normally expect that either A will dominate or B will dominate, right? This is what normal Mendel told.
Yes, sir.
But instead, we see that both are expressing equally. And they are not getting blended. They are not getting affected by each other. So if one is, both are expressing to different levels and both are getting blended. Okay. So the whole flower, instead of being red or white, is now pink, that is blending. But if I tell you that a flower has 6 petals, 3 are pure red, 3 are pure white, what will you call it? Is it incomplete dominance?
No.
It is like dominance or codominance, right?
Yes.
Because both are getting expressed. So here also, the blood group is neither A nor B, it is AB. This is the fourth type of blood group, where in a heterozygous state, no single allele is able to express itself and silence the other. So they are called codominant. They are together dominant with each other. Make sense?
Yes, sir.
Yeah, Mamuna, clear? So these are the two exceptions of law of dominance. And as you said, law of segregation has no exceptions. Now coming to the third law. So again, I'll ask Mamuna. Mamuna, what do you think are the exceptions of law of independent assortment?
Like linkage?
Yes, perfect. Linkage. Now, what do you mean by linkage? First, tell me what does law of independent assortment tells us? Then only we will be able to understand linkage.
Like, is it like something like two alleles will be like differently of different genes will get sorted into gametes? Like that?
Yes, you're perfectly correct. So not two alleles, but here it talks about two genes. Okay. So two alleles of the same gene gets segregated. That is law of segregation, right?
Yes.
Law of segregation says that this capital T small t, even if an organism has capital T and small t, when this organism will go on to make gametes, this capital T will always segregate from the small t. This will go in one gamete and this will go in other. There will be no blending of in the gamete. So the gamete will not contain any allele which will give a pink color, to be simply. So even in organisms that are showing a pink color flower, their gametes will only contain either capital R or small r, one. And capital R will be for red and small r will be for white. So their babies can still show either red or white flowers. It's not necessary that one, because they have a pink flower, they have blending. So their babies also will have blending, not necessary, because that happens through segregation of alleles, purity of gametes. So simply it means that gametes are always pure, even if after fertilization they start blending, but again when they will be formed as gametes, they will be pure. Correct? Do you understand, everyone?
Yes, sir.
But law of independent assortment says that one gene or one character is independent of the other character. For example, your eye color is independent of your hair color. Both are color genes, but it's not like if someone has a black eye color will also have a black hair color. Okay. But Mendel thought that every time it will be true that these genes are independent. Someone can have black eye color with a brown hair color. Someone can have brown eye color with a black hair color. And it will always just like the alleles of the same gene, two different genes will also segregate independent of each other. But that was not true. And for the first time, T.H. Morgan found out in Drosophila, in fruit flies, that one gene can be associated with another gene in a physical manner. That is called linkage of two genes. So wherever that one gene will go, the other gene will also go with it. Remember that? That's called linkage of genes. Sometimes two genes are linked together, and that is an exception to the law of independent assortment. Okay. This here. linkage. Yes, linkage. Okay. So he discovered that the gene for yellow body is linked with white eye, and the gene for brown body is linked with red eye. So more than 90% of the progenies which have yellow bodies always have white eyes, because the linkage is not 100%. It is still, if the linkage is 95%, there will be 95% of the progenies with the same linked genes. 5% there you will still find some recombinants. Depends on how much the genes are linked. What is the percentage of linkage? If two genes are 100% linked, all the progenies will always get it together. They cannot segregate. So there cannot be a assortment. Is it clear, both of you?
Yes, sir.
So in the next class, I will take from